Hi, my name is Nadia Maji, and this is my poster presentation on microhabitat selection in sandfly larvae, with preference to different types of rearing material. For a quick introduction, sandflies, specifically Phlebotomus papatasi, are commonly known as being natural vectors of Leishmaniasis, but they also transmit bacterial and viral pathogens. Leishmaniasis still affects approximately 0.9 to 1.6 million people annually, and it is ranked among the top 10 neglected tropical diseases. Due to cost and access limitations, the availability of treatments in some second and third world countries is limited, and there is no vaccine at this moment. Therefore, it is important to reduce the exposure through population control. Sandflies themselves have 4 larval stages: first, second, third, and fourth instar. In this study, we looked at first, second, and third instar larvae, with fourth instar being in the future. For our research questions, we were questioning whether sandfly larvae have a capability of adjusting their location within the oviposition sites to locales containing appropriate food. And if they have this capability, are they attracted to one medium over another? For a little bit of background information, there's very little research done on the habitat preference of the larvae themselves. It's currently kind of unknown the role that sandfly larvae play in habitat selection after oviposition and hatching. Previous studies showed that adult gravid female sandflies had a preference for second, third instar medium. For our hypothesis, we hypothesized that the larvae will have the capability to adjust their location within the oviposition site to one with food. And due to the previous study that showed that the adult gravid female sandflies preferred the second, third, we also hypothesized that the sandfly larvae will take after the mother and show preference towards the second, third instar medium as well. For our medium, we used 4-ounce Nalgene jars with plaster-of-Parris base, environmental chamber, first and second, third instar Phlebotomus papatasi larvae, water as the control, and the 3 mediums of choice, which included fresh larval feed, second, third instar medium, and along with expired larval medium. So we prepared 12 Nalgene jars for each of the 3 mediums, with 10 first instar larvae in the center. 2 to 3 drops of the control, in this case water, was placed on the side labeled C for control, and 2 to 3 drops of the medium were placed on the other side labeled T for treatment. The jars were then placed in an environmental chamber for 24 hours approximately, and then viewed at 1.5 under a microscope, and the results were recorded. This was repeated for the second, third instars, except instead of 12 Nalgene jars, only 10 were done for each of the 3 mediums. Our results showed that there was no significant preference for one food type over another in either the first or second, third instars. Although with a larger sample size, this might change a little bit, might have a higher significance. There was definitely a clear preference for the locales containing food over the control, with being significant in all 3 instars, with the p-value being less than 0.05. The larvae showed that they have the ability to adjust their locations to the locales containing food, although there was no significant preference for one food type over another. This suggests that while the larvae have the ability to adjust their location, there is either no difference in the food mediums, or the larvae themselves do not have the ability to recognize a difference in the food. For our future work, we are going to be finishing with the 4th instars. We're going to reproduce the experiment with all 4 instars again to include a larger sample size. Another student in Dr. Wasserberg's lab is also looking at rearing larvae in different mediums to see if there is an effect on their growth. This will go back to these suggestion where it suggests that the larvae there might not be a difference in the food. If there is a difference in the food, then that shows that the larvae just cannot recognize the difference.